Alright, so let's talk about photosynthesis. This is the process by which plants utilize energy from the sun to create energy that they can use, largely in the form of glucose and other derivatives of glucose that form all the building blocks of plants. So the process is the combination of CO2 from the atmosphere with water to produce glucose and the release of oxygen. So the process can be limited by light, as you're probably aware, but it can also commonly be limited by the abundance of CO2 in the atmosphere, and we'll talk about that a little bit more. Towards the bottom of this slide, you can see the equation for photosynthesis, and it's useful to note that it takes 6 molecules of CO2 in order to produce 1 molecule of glucose. So this video is all about understanding the different stages of photosynthesis, where those stages occur and what they involve. And photosynthesis is divided broadly into 2 sets of reactions. The light reactions, these are the reactions that require light in order to occur, and then the dark reactions, which can occur in the absence of light. So the light reactions involve 2 stages. The first is the capturing of energy from sunlight by photoreceptors or photopigments, most commonly chlorophyll A and B, but there are also secondary pigments like carotenoids. And then the second stage is the production of ATP and the reduction of NADP plus to NADPH. So ATP and NADPH are high energy molecules that are going to fuel the dark reactions. And the production of those ATP and NADPH molecules occur through photosynthetic phosphorylation. So the dark reactions, this is essentially carbon fixation, so the use of ATP and NADPH to synthesize organic molecules like glucose. So the sites of photosynthesis are chloroplasts. So these are membrane-bound organelles, and they contain within them internal membranes called thylakoid membranes. And those thylakoid membranes at certain points form stacks, and these are called grana stacks, and you can see these throughout the chloroplast. And they're connected by just kind of single sections of the thylakoid membrane called stroma thylakoids. And it's on all of these membranes in the grana stacks and on the stroma thylakoids that the light reactions of photosynthesis occur. The dark reactions of photosynthesis occur just in the stroma, which is the liquid matrix of the chloroplasts. So what this diagram is showing is the light reactions of photosynthesis. So all of this is happening on the thylakoid membranes in those grana stacks and on the stroma thylakoids within the chloroplasts. Essentially what happens is that photons of light are captured by pigment molecules. And those pigment molecules occur in kind of groups called photosystems. So when a pigment molecule is excited, it passes that excitation energy between adjacent pigment molecules. And eventually that excitation energy finds its way into the reaction center. So this is the reaction center of photosystem 2. This is the reaction center of photosystem 1. So an excited electron is then ultimately passed to an acceptor molecule, and these membrane proteins within the thylakoid membrane will then pass electrons between each other, and some of them will pump protons, across the thylakoid membrane, creating a proton gradient. And when those protons flow back across the membrane, they do so through ATP synthase. And so ADP is phosphorylated to produce ATP. So this is the first high energy molecule produced by these light reactions of photosynthesis. The final electron acceptor as part of photosystem 1 is NADP+. And so by accepting an electron, that molecule is reduced to NADPH, which is the second high energy molecule produced by the light reactions of photosynthesis. So the production of NADPH involves the loss of 2 electrons. And so those electrons lost by photosystem 1 are replaced by photosystem 2. And that occurs through the cleaving of water molecules. So for every 2 molecules of water that are cleaved, electrons are released and one molecule of oxygen is released to the atmosphere. So the oxygen that's released to the atmosphere as a result of the light reactions of photosynthesis occur through the breakdown of water, not through the breakdown of carbon dioxide from the atmosphere. So these are the different stages of the light reactions of photosynthesis that I've just described. So first of all is the primary photo event. That's when a photon of light is captured by a pigment molecule. Then you get charge separation, which is essentially the movement of that excitation energy between pigment molecules and ultimately into the reaction center. Then electron transport, that's the exchange of electrons between proteins on the thylakoid membrane, resulting in proton pumping and the generation of a proton gradient. And then finally chemiosmosis, which is protons flowing back across the thylakoid membrane through ATP synthase, generating ATP. So we're moving on now to the dark reactions of photosynthesis. So this is the Calvin cycle. So this is the reaction that doesn't require any light in order to function, but it does require these high energy molecules, ATP and NADPH, that have been produced by the light reactions of photosynthesis. So they're the energy that drives this cyclic reaction. And there are multiple cycles of this reaction that are needed to generate a molecule of glucose. So really the first thing that happens in the Calvin cycle is CO2 is combined with a molecule called ribulose 1,5-bisphosphate, which we happily can shorten to RUBP. And the combination of those two molecules is catalyzed by this enzyme Rubisco. So Rubisco is an extremely important enzyme and one of the most abundant proteins on the Earth. But it is for some reasons slightly inefficient, and we'll come on to talk about that in a little bit in a few slides' time. When RUBP and CO2 are combined, they produce a 6-carbon molecule, which is immediately broken down to form 2 3-carbon molecules of phosphoglycerate. So the first kind of stable product of the Calvin cycle is a 3-carbon molecule, and that's why most photosynthesis goes by the name C3 photosynthesis. That's the most common form of photosynthesis. So for the remainder of the Calvin cycle, a set of reactions catalyzed by or powered by ATP and NADPH produce 6-carbon molecules, glucose and fructose, and polymers of those molecules like starch, cellulose and sucrose go on to form all of the building blocks of plants. Then in the remainder of the Calvin cycle, this molecule, this 5-carbon molecule of RUBP is regenerated so that the cycle can start again. So really the phases of the Calvin cycle are carbon fixation, then generation of organic molecules like glucose, and then regeneration of RUBP. And it takes 6 cycles of this Calvin cycle to produce 1 molecule of glucose. So as successful as the process of C3 photosynthesis is, it does have its limitations, and those limitations come as a result of the inefficiency of that enzyme Rubisco. So the reason that Rubisco is slightly inefficient is that as well as catalyzing the reaction between CO2 and RUBP in photosynthesis, it also combines with oxygen molecules in a process known as photorespiration. So any Rubisco enzymes or proteins that bind to oxygen rather than CO2 reduce the rate of photosynthesis in the plant. And this is a particular problem in warm environments. The reason for that is that in warm environments, plants may have to close their stomata in order to preserve water. So they lose a lot of water through the stomata, through the process of transpiration. But it's through opening stomata that plants actually allow CO2 from the atmosphere into the plant. So when stomata are closed, oxygen generated by the process of photosynthesis builds up in the air pockets within the leaves. And the concentration of O2 compared to CO2 gets higher and higher when those stomata are closed. And so as that happens, rates of photorespiration get higher and higher, and rates of photosynthesis get lower and lower. But there are some plants that have evolved a way around this problem, and there are two different, more recently evolved types of photosynthesis that can, to some extent, get around this problem. And these are C4 photosynthesis and CAM, which is Crassulean acid metabolism. And I'll explain in a bit more detail what the difference between those two types of photosynthesis is. Before I do that, though, I just wanted to talk a little bit more about the process of photorespiration. So this is a competing process with photosynthesis, essentially that occurs when the enzyme rubisco binds with oxygen instead of CO2. And so photorespiration is generally seen as a negative thing that competes and reduces the rate of photosynthesis. But it is thought that photorespiration might have some benefits, specifically protecting photosystem 2 from damage that can be caused by an excess of electrons. So the alternatives to C3 photosynthesis are C4 and CAM. And both of these different styles of photosynthesis use a different enzyme, PEP carboxylase, instead of rubisco. And rather than producing a 3-carbon intermediate, phosphoglycerate, they produce a 4-carbon intermediate, oxaloacetic acid. And so this 4-carbon intermediate is what gives C4 photosynthesis its name. Fundamentally, the difference between C4 photosynthesis and CAM is that C4 separates the light and the dark reactions of photosynthesis spatially. So the light reactions occur in mesophyll cells close to the surface of leaves. The dark reactions occur deeper in leaf tissues in bundle sheath cells. CAM photosynthesis temporally separates the capture of light and the uptake of CO2. So essentially, CO2 is taken up during the night when stomata can remain open with relatively low water cost to the plant. So what this diagram describes is C4 photosynthesis. So you can see ultimately that the light reactions occur in mesophyll cells close to the surface of leaves. And the dark reactions occur in bundle sheath cells, so deeper within leaf tissues. Carbon can be stored in organic form in the form of malate in 4-carbon molecules. And that malate can then be stored in bundle sheath cells. And CO2 can be liberated from malate as it's needed for use in the Calvin cycle. So the advantage of this is that within these bundle sheath cells, concentration of CO2 can be kept relatively high, and it doesn't have to compete with oxygen for binding sites on the enzyme rubisco. So there are thousands of species worldwide that use C4 photosynthesis, but it's an adaptation really for warm climates. And the most common groups of plants that use C4 photosynthesis are grasses from warm grassland habitats. And one example is this species, which is sugarcane. CAM plants, or Crassulean acid metabolism plants, tend to be very succulent plants growing in extremely dry environments. And I'll come on in a second to kind of explain the reason why that is. So this diagram explains the process of CAM photosynthesis. So essentially what happens here is that these plants in their very dry, arid habitats will only open their stomata at night. And that's because opening stomata in the day would lead to too high water losses through transpiration. So during the night, that's when these plants absorb CO2 and they turn it into an organic form in which they can store it, which is malic acid. And they'll store these malic acid carbon molecules in large vacuoles. So that's why most CAM plants are very succulent plants, because they need a lot of space, a lot of large vacuoles where they can store malic acid, because the amount of malic acid they store controls essentially the rate of photosynthesis that they can achieve. So CAM photosynthesis is very much a temporal separation of the dark and light reactions of photosynthesis. And that's because the absorption of CO2 can only happen at night. So CAM plants make their sugars during the day with CO2 that's been absorbed during the night. But one other advantage that CAM plants have is that some of them, when conditions are okay for stomata to be open during the day, they can switch to C3 photosynthesis. So they can be facultatively C3 plants. And so this is a big advantage for those species that can rotate between those two different types of photosynthesis. So this is what a typical CAM plant might look like. This is a cactus, Opuntia maxima. And as I mentioned, most CAM plants are very succulent because they have to store large vacuoles so that they can store high quantities of malate. But CAM really is an adaptation to extremely dry environments. So the kind of plants we might imagine are like the cactus that we just looked at. But actually, epiphytic plants often are CAM plants. So these are two species that can be found here in South Florida. And so you have to imagine that even though here in South Florida we live in a relatively wet climate, there are still microhabitats that exist that are extremely dry, arid environments. And so plants, epiphytes that actually use other trees or plants for support, can find themselves in very dry microhabitats. And so for the same reason as cacti might use CAM photosynthesis, so do these epiphytic bromeliads. So I wanted to spend the last minute or so just looking at the different photosynthetic pigments that plants have. So what you're looking at now is the spectrum of visible light. So this is essentially white light divided into its different constituent wavelengths. So those wavelengths span from about 390 nanometers to about 750 nanometers. And what this white line shows is the efficiency with which chlorophyll A and B absorb light of different wavelengths. So you can see they absorb quite a lot of the violet and blue light. They reflect all of the green light or most of the green light. And then they absorb efficiently again much of the light in the red band. So the fact that they reflect green light obviously tells us why we perceive plants to be green. But as well as chlorophyll A and B, plants also have secondary pigments, most commonly carotenoids and lutein. And carotenoids in particular absorb a fair amount of the blue and violet bands, but really their role is to absorb light from a broader range of wavelengths, albeit at a lower level of efficiency. When deciduous plants start to reabsorb their chlorophylls and drop their leaves at the end of summer, the beginning of winter, that's when we see the colour of these carotenoids coming through in those fall leaves.